I think we can get started if you guys are fine. So welcome to this 7th lecture on the intermediate course of SynBio4All. My name is Enrico, and today we will speak about the applications that we can have for microbes for sustainability. So you might not know who is this guy that is speaking, so I would like to just give you a brief bio about myself, so you know what are a bit my expertise. So I'm Italian, probably you guessed it from the accent. I was born and raised in Italy a bit more than 30 years ago, and I studied biotechnology first in Italy, and then I moved to the Netherlands. And there I got a PhD in metabolic engineering at Wageningen University. And since then I moved a bit around between mainly Europe and US, keeping on doing research. And at the moment I'm based in Copenhagen in Denmark. So yeah, in Scandinavia, in Northern Europe. And I'm doing a postdoc, so I'm keeping on doing research, and my interest, so what drives me, my motivation, what makes me excited when I wake up in the morning, is to try to learn how bacteria function and how we can engineer them to use these organisms as cell factories for bioproduction. And in particular, I'm highly interested in ways in which we can take CO2 from the atmosphere and convert it into useful products. So it's this field of CO2 valorization. You will see we will also touch a bit on that during the class. So today we will, as I said before, work on we will work on some take-home messages on on the topic of microbes for sustainability. So these are the take-home messages that I would be happy if we digest throughout the lecture and you bring with you when you go to sleep. So the first thing is I want to emphasize that the microbial world is very diverse, and there are a lot of metabolic traits that we can experience and we can observe as scientists. These different metabolic traits give us opportunities for developing sustainable processes. And current century, so the 21st century, will probably be, and we are already seeing it, it's the century where biotechnological applications are getting into place. And we will see many applications also in the field of microbial biotechnology. I hear some sound. Okay. No question. So, and then the other point I wanted to raise is that, to support this bio-based transition, SynBio, which is the content of this course, is one of multiple aspects that need to, work synergistically together, including metabolic engineering and also process engineering. And when we look at synthetic biology and looking at improving microbial traits, we can follow two approaches called top-down or bottom-up. And you will see examples from both parts. So today, yes? Did someone ask something? No, I saw, yeah, I heard the sound, but it was not the case. So about the outline of today. So what will happen, we will first touch on general notes and concepts, just to give you a bit of, understanding. So we will dive into the overview of what microbial diversity is and what are the different trophic modes. And with trophic modes, it means the ways in which microorganisms can grow and how we classify them. Then I would like to touch on the design, build, test, and learn paradigm for strain engineering. If I am not mistaken, you must have seen this already in one of the previous classes. Then this top-down and bottom-up approaches for strain engineering, how they complement each other. And then I would like to touch on the UN Sustainable Developmental Goals, which are some guidelines on how to improve society as a whole. And we will use this as, ways to contextualize some areas where microbial biotechnology can tackle and improve sustainability. And so in the second part of the class, we will dive into some of these examples. A disclaimer from my side, the examples I will show you are the ones I'm aware of, that I have experienced, like through either direct contact with people or by reading research literature. But there are plenty of other examples that exist that I might not be even aware of that will not end up in this class. So with this in mind, I think we can start diving a bit into the microbial world. So what you see here is it's a tree of life. And do you know what this tree represents? Does someone want to say what we are seeing?
Does it represent the 3 domains of life?
Perfect, yes. Thank you, Billy. Mm-hmm. But what do you see listed here? What are these names? And
phyla of bacteria.
Yeah, so indeed, what we see are branches of this tree, and at the end of each branch there is a certain taxonomic class and category, and it shows that all have a common origin and they are diversified into different domains. Do you guys know by any chance an example of a bacterium? Yes, I see Nelson writing E. coli. Perfect. Pseudomonas. Perfect. Moving to eukaryotes. So I see some names. I think most of these are bacteria. Saccharomyces cerevisiae, yes, this is a yeast. And there is an A. I think is Valerie. Yes. And so let's say eukarya have a lot of species. We also are eukarya. So the difference between bacteria and eukarya is the compartmentalization. So eukarya are a bit more, let's say, complex in their morphology and what they contain. Are you guys aware of what archaea are, for example? So if no one knows, if no one says it, so archaea, it's another type of prokaryotes, so meaning organisms that do not have any nucleus, and they are a bit more exotic. They are something in between bacteria and eukarya, and it seems actually that originally the first eukaryote that ever emerged came from a bacterium that entered into an archaea. So let's say it's also a prokaryote that often grows in weird environments and are actually interesting organisms to study that are a bit yes, exotic. I see also Sarah indeed commented that archaea live in extreme environments. That's true. But so this tree of life, it's divided into these 3 domains, and all include microbes. So yeah, as you have seen, like we mentioned, for example, in eukaryotes, Saccharomyces cerevisiae, that's also a microbe. So the microbial world is represented in all 3 domains of life. And when I talk of microbes, just for being everyone on the same page, I talk about unicellular organisms. So we talk about this wide diversity that there is in nature and the presence of unicellular organisms a bit everywhere, but I also wanted to quantify this a bit, and I think this graph helps a lot. So this is a square that includes all the biomass that is on on our planet. So if we look at the amount of carbon that is present, the vast majority of it is in the form of plants. So of 550 billion tons of carbons that are there, 450 are in plant biomass. So this is 80% of it. But the second biggest amount of carbon is in living organisms belonging to the world of bacteria. So there are plenty of bacteria outside in the environment. And also archaea, they are much more than, for example, all the animals that are on Earth. And we as humans are a very, very tiny amount. So if we make just the sum of bacteria plus archaea, we have basically about 15% of all the living biomass. So there is plenty of microbial biomass existing in the world. And yeah, we have seen that they are pretty distributed, and you know, they can thrive on many different environments. So we can find them, for example, at the bottom of the ocean. That's a place where we have these alkaline or hydrothermal vents. That's a place where, for example, we would find a lot of archaea. We can find also organisms growing in volcanoes, in lakes that can be for fresh water, or also with also some sort of extreme conditions like salty lakes, soil, also glaciers. So we really see that we can span through many different ranges of temperatures. And also, for example, in our gut, from poop, as you will see in the future, in some future next slides, is also a source of interesting features for biosustainability. So yes, microorganisms are indeed ubiquitous. For classification purposes, it's important to be able to describe how these microorganisms can grow. So there are some terms that we use, and these are based on you know, some questions that we can ask ourselves, and this will tell us how microorganisms grow. So the first thing is look at the source of energy. So how do organisms obtain their energy? Where do they get their energy from? In case they get it from chemical sources, we call these organisms chemotrophs. And on the other hand, if they get this source of energy from light, we call them phototrophs. Then the other type of aspect we look at is where they get their carbon source. Meaning, yeah, do they get CO2? Do they eat organic carbon? So in case of a chemotroph that grows on organic carbon, we say a chemoheterotroph. Whereas if it's a chemotroph that can assimilate CO2 as carbon source, we talk about chemoautotroph. On the other hand, when we deal with phototrophs, if they eat organic compounds, we talk about photoheterotrophs. Or if they eat CO2 using light as a source of energy, we talk about photoautotrophs. So there are also other sub-classifications we can use. So in the case of chemoheterotrophs, what is their final electron acceptor? So if and I think here I forgot to write it down, but if there is oxygen, basically almost all micro and major organisms grow in a chemoheterotrophic regime like us. If they cannot grow using oxygen, either they are strict fermentative bugs or they do anaerobic respiration. Then when we talk about chemoautotrophs, these are like the class of compounds we get, so hydrogen oxidizing bacteria or sulfur or iron oxidizing bacteria. When we talk about photoheterotrophs, green, purple, and non-sulfur bacteria. And when we talk about photoautotrophs, we can also distinguish between the ones that do oxygenic photosynthesis. So they use they strip electrons from water to reduce CO2 and make glucose, and oxygen is a byproduct. Or in case they are not capable of doing that, we talk about organisms that perform anoxygenic photosynthesis. But with this graph, we basically cover the whole type of metabolisms that we can find in living organisms. So now that we kind of have a bit of an overview of the diversity that there is in the microbial world in terms of metabolism, we can start moving into the genetic manipulation. So here I know that is something that you already covered, so we will touch it very briefly. But I would like to refresh a bit what is the central dogma of synthetic biology. Actually, if someone feels like and would like to say it, please go ahead. I'm happy to let you guys speak. Yeah. So indeed, Miriumi got it right. So we have a flow of information that I saw that Salomon also raised the hand, but we got already the answer in the chat. So the idea is that we have DNA that is transcribed into RNA, and then RNA needs to the synthesis of proteins, and these proteins have a function. So assuming that we have a function, let's call it A, this is how the flow of information goes. With genetic manipulation, we can play and we can interfere with this flow, and by doing gene deletions, we can remove DNA and remove information. Or on the other hand, we can add heterologous DNA and provide new information for a new function. So what you see here, function B is removed, function C is added. So when you look at the microbial cell as a platform for biotechnological application, it's nice to being able to program this cell by working at the DNA level. And you have some specification as a designer that you want to have in your bug, and you can do this by adding or removing DNA. And the way in which you systematically perform these modifications and you assess them, it's called the design, build, test, and learn cycle, which is something that is borrowed from engineering as a field. So you see it here. It's a cycle where in the design phase, you kind of check what you want to modify, and you kind of design your experiment. You get you do your in silico analysis. In the build phase, you perform the modifications. In the test phase, you collect the data that tell you what the effect of these modifications are. And in the learn phase, you do data analysis and you refine your original hypothesis, and you see if you can start a new cycle. And machine learning can become handy to really close the gap between learn and design and accelerate this part.
Enrico, there is a question in the chat.
Yes, I see Sarah is asking if we can have proteins back to RNA or not yet researched. Can you elaborate more? What do you mean with the question? So if the flow can go in the opposite direction. So I'm not aware of proteins that. So let's say there are definitely ways in which this loop can be reversed, but I must confess it's not really my my field of expertise. So I would say that it's possible that some proteins can have an effect on RNA. And for example, there are some of these Cas tools that are capable on working on already transcribed proteins and change their fate, so to say. But I wouldn't be able to tell you much more than this, to be honest. I don't know if a protein can change its own RNA in the reverse direction. So unfortunately, I think I'm not capable of giving you the answer you wanted. So Yeah, related to the DBTL cycle, and anyway, sticking to this dogma of synthetic biology, which is, anyways, can always be, put under discussion, and I'm sure there will be exceptions to this dogma existing. This is how we, formalize the pipeline for strain engineering. So these DBTL cycles that you have in place can be operated in two ways, either a top-down approach or a bottom-up approach. And I would like now to spend a few words to kind of differentiating them. So the top-down approach, it's all about streamlining what are native characteristics of the organism. So a typical pipeline for top-down approach starts with bioprospecting, meaning isolating strains from the environment, get pure strain isolated, then characterize these strains, and then once you have the strains that are in place and you know how they behave, we can develop tools for genetic manipulation that allows us to get mutants, and these mutants have an improved phenotype. And this is one way to do synthetic biology. The other approach is called bottom-up. It's more about building up the features that you want in a surrogate host. So how this works, you don't take strains from the environment, but you start with a model organism that already has quite advanced genome editing tools. And since you know this organism well, you can also do more daring engineering approaches and, perform heavy engineering on the strains and really deeply rewire the metabolism, and maybe use also evolution, and you get to, again, an ideal strain that has the features that you want. Both approaches have pros and cons. So when looking at the top-down approach, advantages is that you do not need to have a deep knowledge on the bug. So you don't need to have the whole genome that is sequenced, for example, although now it's relatively cheap to do so, but you in principle do not need all these informations as start. And how the top-down approach mostly works is in a reductionistic approach. So you remove things, which is in general easier than adding. And it's something that allows you also to maybe enhance some features that are already present and that nature has selected for in billions of years of evolution. The disadvantage of this approach is that you might be a bit restricted in the design and solution space because you do not have a lot of information. And generation of mutants can be difficult because you might not have like the state-of-the-art tools. And the lack of extensive information can slow down also your pipeline. On the other hand, the bottom-up approach has the advantage that you really kind of design the metabolism as you want. So you, in principle, at least in theory, you can exert more control on the metabolism, and you can apply a higher design space of solutions. So you can plug in pathways that are not there, for example. And this allows you also to challenge nature and find other solutions that maybe evolution didn't select for. And so this is also quite stimulating, I would say, as an approach. The disadvantages is that often these strains are very slow at growing because they have a lot of mutations. And you also need a lot of prior knowledge on the metabolic network of the organism and the genome and everything that is in there, because you need to build starting from this information. And overall, this approach has a higher chance of failure. So now I would like to do a small quiz, just for, as an intermezzo. So we saw before the different domains of life. So I just wanted to have a feeling of how many people are following. So the question here is, which domain of life includes unicellular organisms? So I will list them down, and then in the chat, you guys can write the answer, So option 1 is archaea. Option 2, bacteria. Option 3, eukarya. And option 4, all of the above. So I give you like 30 seconds to think and write your answer. Okay, so I think many people followed. I think the majority selected for option 4, and I think it's the correct one, because as we said before, all of them can include unicellular organisms, and we listed also Saccharomyces cerevisiae as an example of an eukaryote, which is unicellular. Okay, second question. So now reset, focus. So I wrote it wrong. Sorry, I formulated wrong. But the question was what does DBTL stands for? So what is the acronym of? Design, bring, take, look. Design, build, test, learn. Dare, build, test, learn. Dominate, boot, think, load. So also here I will give like 20 seconds. You can choose your answer. Okay, yes, I see. Most of people, yeah, okay, 2, 2, 2. Yes, I think most of you guys got it right. Is design, build, test, learn. Perfect. Now this is a bit more tricky, so I will. So I will now, let's say for this one, I will give you a minute. It's a bit more difficult. So we talked about top-down and bottom-up synthetic biology. You haven't seen examples yet, but based on what we discussed, what is the feeling? What could be an example of a top-down approach in SynBio? Option 1, you take a model organism and you implement a new substrate assimilation pathway in the organism. Option 2, you build a library of ribosome binding sites to control the expression of a heterologous gene. Option 3, you develop a genome editing tool for a newly domesticated bacterium and you use it to remove competing metabolic pathways. Option 4, you perform adaptive laboratory evolution for increasing the growth rate on a native substrate. So this I will give you a minute. And if you have questions or like doubts, please also shoot and we can discuss them. If they are complicated questions, we keep them for the end of the class. Okay, I see different here. Here people are a bit more spread in the answers. So I see some 1, some 3, some 2. So maybe it's not clear that much yet what top-down means. Here I put it. I made it a bit tricky because actually 2 options were correct. So the first one is like, you develop tools in a bacterium to remove competing pathways. Is this type of reductionistic approach that we discussed before. And the other one is, you use adaptive laboratory evolution to improve the organism in doing something that is already capable of doing. So in my view, at least, this also is a top-down approach. We do not add other stuff, but we try to streamline what is already there. Okay, but I think you will see some examples of this later. So I hope you will be this will become clearer also at the end of the class. Okay, so now we move towards the part that is a bit more hands-on on examples. And I would like to start with these sustainable development goals. I don't know how you guys are familiar with them, but in principle, it's a set of goals that was developed by the United Nations. And you see a quote, they are considered a call to action to, fight inequality, poverty, protect the planet, and, ensure health, justice, and prosperity. So it's like all the things that the mankind can do to go into this type of direction. So they are 17, and we talk about no poverty, zero hunger, good health and well-being, quality and education, gender equality, clean water and sanitation, affordable and clean energy, decent work and economic growth, industry innovation and infrastructure, reducing inequalities, sustainable cities and communities, responsible consumption and production, climate action, life below water, life on land, peace and justice, and partnership for the goals. Of course, this includes everything that the mankind has to do for all aspects of society. What I would like to stress is that many of these sustainable developmental goals can be tackled by using microbes. So some examples. So microbial proteins touches the second sustainable developmental goal, so the one about zero hunger. Pharmaceutical biotechnology, it's about the number 3. Wastewater treatment is about the number 6. Using new type of, let's say, performing fermentation of using new feedstocks that come from renewable electricity touches on the sustainable developmental goal number 7. Biomanufacturing as a whole touches number 9. The use of alternative microbial feedstocks in general is about the the SDG number 12. Bioremediation helps for life below water. And number 15, it's like, smart use of feedstocks can avoid deforestation and help with the SDG number 15. So, one second that so what we will do for the rest of the class is to touch on some examples that cover like one or more of these aspects. So we talk about the use of microalgae as a mean to fight, to tackle waste, let's say to enhance wastewater treatment, and their use as a protein source for, let's say, supporting life in a small island. I will talk about the use of, the use of microbes for the conversion of lignocellulosic feedstocks in biomanufacturing. That touches SDG 9, 12, and 15. Then I will talk also about how Escherichia coli has been converted into an autotrophic organism growing on the Calvin cycle. So this also touches 4 SDGs. And then I will touch this a bit more briefly about the degradation of PET plastic in the ocean. This is clearly related with life below water. And also how, now the forefront of pharmaceutical biotechnology allows to produce very complex molecules in yeast. So I will show you the example of vinblastine, that is an anti-cancer drug. Okay, so now we can move on the use of microalgae for SDG 2 and 6. So microalgae, you can see represented here, you should imagine them as very small trees. So it's like unicellular trees or plants that can perform photosynthesis in just their small cellular environment. So they are interesting bugs to work with, and here you see some of their features. So they're interesting because they can grow on salt water. So you can, in principle, grow them on seawater, and you don't need to use fresh water for their growth. Not all species, but some of them, yes. As I mentioned before, they use sunlight as a source of energy. So they can do photosynthesis, and photosynthesis is, the use of light as a source of energy for converting CO2 and water into oxygen and sugars. Microalgae, as I mentioned before, are like small plants, but they have a much higher biomass yield than conventional crops. So per hectare of land that you occupy, you can produce much more biomass with microalgae than with crops. And also, they are sources of high-value compounds like pigments, antioxidants, and omega-3. And what I wanted to write here is that they can be also a source of protein. And here I just copied twice the text. So let's move to these two examples. So before diving into the examples that I wanted to show, I would like to just briefly touch on the concept of bioprospecting. The bioprospecting is like the very first step of these top-down approaches that we discussed before. So how bioprospecting works is that you go into the wild and you collect samples from a place that you think that there are the features you are looking for. So it could be from a volcano if you're looking for something that is capable of growing at high temperature, from a pile of compost, or you can go in the depth of the ocean if you want something that resists to high pressures, and so on. Then you take these samples that are composed by very mixed communities, and you awake them on a rich medium. So you try to bring back to life everything that is in there. And then you move into some selective conditions. So you transfer this mixed population in liquid, and you start applying a selective pressure. And for example, if you want to isolate a microalgae that grows at high temperature, you will use two things: sunlight, and you cultivate at high temperature. Of course, you should also use the type of medium that is good for a microalgae. So it could be, for example, seawater, and could be also without any carbon source, because we saw from before that microalgae are photoautotrophs. And then what happens is that through time, if you look at this graph, this is like the typical graph of microbial growth. This is time. We plot time versus the logarithmic scale of cell density. So we have an initial lag phase. Then you have the logarithmic growth. This is high. Let's say the more the organism is happy in this environment, the higher will be the growth rate. And by doing serial dilutions, we will really enrich our population with these isolates of strains that behave as they should. So this is a great tool in the hands of microbial biotechnologists to start isolating strains that have a good feature. So why we want to do this is because often nature already has a solution for the problem we want. So it's not just the need of being able to select what we need and select with the right parameters. And sometimes the strains behave so good that you do not need to do a lot of synthetic biology on top of them. And also, as you will see, especially in this case of microalgae, especially because we're interested in food consumption, it's better to limit the type of genetic manipulation we do, because this can have an effect also in terms of legislation of what you're capable of selling or not. Applications of microalgae for sustainability. I will show you two examples. One, it's about cleaning wastewaters in dense municipalities in India. And another one is the cultivation on site of microalgae as a source of protein in a small island of the Caribbean, which is called Bonaire. which is part of the Dutch administration. So algae, as I was mentioned before, can grow on sunlight. They do not need carbon, but they need also other components to build up their biomass, especially phosphorus and nitrogen. These are also big elemental chunks of microbial biomass. And a source of phosphorus and nitrogen is what we call the black waters. So these are like the type of water you have, after you flush your toilet, or like, in this biological, how do you call them? Like in the countryside, in Italy, for example, we have these huge containers that you just basically all your secretions go there and they stay there, and every now and then you need to remove them. But let's say they are a great source of like these nutrients that can be absorbed by microalgae. And how this works is as follows. So you have these black waters, and you use them to feed a bioreactor, so a contained environment where you have your microalgae that are isolated locally. And then these microalgae will be exposed to sunlight and CO2. So they will convert CO2 into sugars, and then they will start uptaking nitrogen and phosphorus to build their own biomass. And what will happen is that through time, all these nutrients will be taken up by the algae. The water will become clean, and by applying some filtration, we will be able to split the biomass, the microbial biomass that is obtained from the fresh water that is now removed from the waste that was present made by human feces and urine, whatever. And this fresh water can be used back for example, irrigation of crops. Whereas the biomass, the algal biomass that is obtained, because it's full of nutrients, can be used as feed source for animals, for example. So this is a way to clean up water and apply some concepts of circular economy. The other example comes from hands-on experience of some colleagues of mine that during their PhD, they had this challenge of supporting the build-up of a protein factory in this small island in the Caribbean called Bonaire. And there they wanted to use local algae that can grow at high temperatures and that they can be rich of proteins. So there was this guy, was a PhD student, that at the beginning of his PhD put his hat on and went to the Caribbean and started sampling from different ponds in the island, where he saw some, parts that were under sun that had high temperatures. He got some samples, he went back to the lab, and he started applying this concept of bioprospecting. So he started to do serial dilutions of these complex communities using also high temperature as a selective pressure. And what he managed to obtain was to isolate eventually some strains that were useful for his purpose. And now I will show you a bit also these results. But what I wanted to highlight is that these two types of application can really create, as I'm seeing also in the chat, people commenting, a real circular loop of resources. So imagine now you have highly dense cities. People go to the toilet, they do their own things. They throw their waste into these containers that then can be used to feed microalgae. And we can remove all these wasteful products that would be polluting from the water. We can create microalgal biomass with that. And at the same time, this microbial biomass can be used either as food system, or we can extract some valuable products like for nutraceutic that can be used. This type of approach, I see the question of Nelson. I'm pretty sure there are plenty of projects like this, and it's a matter. I think the only problem, as you can see here, is the infrastructure. You need to build big facilities to scale up these cultivations. And what I wanted to show here is that there are different type of reactors that you can envision for your cultivations. The challenge of growing microalgae is that they are very dependent on sunlight. So you see these reactors really try to occupy as much as possible in terms of surface, so you can really have a high irradiation. And they tend to be also very thin because at the center of the reactor you have shadow. So you want to minimize the part of shadow for your microalgae. So that has some logistical, no, not logistical, more process design or reactor design challenges. But once that these reactors are prototyped, then they can be exported where it's needed. Then the other example of the isolated microalgae. So this is all published data, and there are, I forgot to mention, all the things I'm showing here can be backed up by the references. So what you see in here, these are like the strain that were isolated. One of these ones, the ones on the right, has the highest growth rate with the temperatures that are above 35 degrees. So they're really good at growing at high temperatures, and also good enough for the guy that did the study. This strain specifically had also quite high protein content. So 70% almost of its biomass is made of protein. So it's a perfect check for the purpose of the study. And these strains are going to be grown into a new type of reactor that is shaped as V, that has been designed like that because it should be floating on the sea. And because of how the water changes the inclination of the irradiation, it's intended to maximize again the light to which the microalgae are exposed. I see a lot of questions. Since I get distracted, I will not answer them right now, but we can go through them towards the end of the class. But I think they are all very good questions. So please bear with me and keep on writing. But if I start looking at that, then I get distracted. And I cannot do two things at the same time. Then I wanted to move to a second example. So this one is about biomanufacturing. With biomanufacturing, we mean trying to shift from a linear way of producing goods and products. That is what we have here, that is an economy that is based on oil, into a more circular type of economy. So the idea is that we do not use oil as a feedstock, but we would, on the other hand, use a feedstock that can be regenerated from CO2. We will see in a second what do I mean. And rather than relying on chemical synthesis, that is also quite polluting, we would rely on biological conversion. So we will use cells as small chemical factories to perform the conversions we need. This approach in general is considered to be more green. The challenge is to get those conversion rates and performances that can compete with the traditional chemical industry. So there is this line that you see here that is from CO2 to our feedstock, and you see that there is also renewable energy mentioned. Do you guys know which type of renewable energies we deal with? I mean, there is the cartoon there, but I was wondering if you are familiar with which type of renewable energy sources exist and are accessible also technologically.
There is submission in chat.
Excuse me?
There is something in the chat.
Yeah, I see Maxwell writing bioethanol, biofuels, but this is not what I mean. Renewable energy means a source of energy that is present in nature. Yes, so Nelson has written wind, that's one. Light, yes, Salomon is right. Solar. And there is another one that is water, so the hydropower. These hydroelectric factories that are constructed also with dams. Also, they are meant to generate energy. All these are sources to create electricity that then can be used, you know, to make some sort of feedstocks. But in terms of, like, you know, if we want to this light to be like biology, it can be the sunlight that grows crops from which we extract sugars that we use for fermentation. So actually, yeah, how, you know, this biomanufacturing works, the concept is relatively simple. So we have a feedstock from which we extract a substrate. This substrate is thrown into a bioreactor. And in this bioreactor, we have an organism that we call a cell factory. That is a unicellular organism that it's engineered, maybe, you know, expressing a metabolic pathway of interest. And inside this bioreactor, the cell factory takes the substrates and converts it into a value-added compound that, you know, we can use for a bio-based economy. So we were talking about, you know, these feedstocks that can be generated from CO2. Throughout the history of mankind, we've seen that there are at least 4 generations of feedstocks that have been considered for this type of transformations. So the first one is the one of edible biomass that we can obtain from crops like sugarcane or in Northern Europe, beetroots. This is something that has been used since the beginning of agriculture. So, you know, doing bread or fermenting wine. These are like the type of biotechnological processes that mankind has always done since the beginning of times, using first generation feedstocks. But this type of feedstock has the problem that we really consume the edible part of biomass that we would rather use for human consumption. So during the 20th century, so during the last century, it started to, you know, there started to be interest on using the non-edible part of biomass, like lignin, as feedstock for microbial cultivation. That has some challenges that we will see in a moment. But, you know, the focus has shifted, and it's a more, you know, sustainable way of using resources, because we would not compete directly with the food supply chain. Then, especially since the beginning of this century, there started to be a lot of interest into microalgae, as you have seen before, as, you know, ways to perform conversions. And now, this, especially since the last, within the last 10, 15 years, source points that emit a lot of CO2. So imagine, for example, waste pipes, exhaustion pipes from, for example, the steel industry that is full of CO2, are considered as, you know, sources from where to obtain your feedstock. And this part, you will see later, can be coupled with the generation of, with the use of renewable electricity to put electrons into CO2 and generate what we call C1 feedstocks. Okay, so I was mentioning, I would like to dive into the example of lignin valorization. So this is what we are touching now. So as I mentioned before, lignin is not the part of the plant that we would eat. Nevertheless, it's full of carbon, and it's full of carbon that in principle can be used by microorganisms. So if you look at a crop and you look at the plant cell, there is this part that is called the cell wall that is filled with this microfibril, which are you know, complex structures that are made of hemicellulose, lignin, and cellulose. And especially cellulose and hemicellulose, they're made out of sugars, glucose molecules, or pentoses and hexoses. And they are a significant amount of, you know, this non-edible part of microbial biomass. So compared to the traditional pipeline for using first generation feedstocks, which relies on the edible part of the biomass that you see here, we will have, for example, sugar cane. We extract glucose, and then glucose is inoculated into a pilot reactor and then scaled up together with the organism. If we want to use lignin, we need to do some pre-treatment. So we need to make these monomers readily available for the cell, and this has a cost. We need to add enzymes, what are called saccharolytic enzymes. These enzymes work at high temperatures, so we need to heat up, these pots, have the enzymes working at high temperatures. But then if we work with organisms that grow at 30, 37 degrees, which are called mesophiles, we need to add energy to cool everything. And then we will have this mixed and complex media containing glucose, which is a 6-carbon molecule, xylose, which is a pentose, and other aromatics that you see here. And then we need to inoculate them and then scale this up. So these steps, let's say, although the valorization of lignin is promising, there are these extra steps that make our operating costs more expensive. And there are then two examples that I wanted to show of how you can use microbes to tackle these problems. So one is about using thermophiles so that we can perform our fermentations at those high temperatures directly after that the saccharolytic enzymes were used. And then I will show a second example that is about using yeast, but then trying to valorize as much as possible this mixedly composed pool of substrates, teaching yeast how to accept also pentoses together with exosis to do the fermentation, and so recovering much more carbon and transforming that into product. So bear with me. Now we will go about the domestication of thermophiles. So this is a work that was done also by some colleagues of mine in Wageningen. So they knew that they needed to have organisms capable of growing around 60 degrees, because that's the temperature at which these saccharolytic enzymes are working. So they went into, the local municipal waste place, and they went to a huge pile of compost. Compost, I don't know if you're familiar on what it is, but it's nothing but organic waste that is piled up together, and you let it, the microorganisms that are there, you let them digest and ideally respire and degrade everything. So at the core of this pile, temperatures are pretty high. They can reach up to 70 degrees. So they went there, they took a sample, and then they started to basically, and they did enrichment in the lab, and then they started to apply some selective pressures. Since they wanted an organism that is quite flexible in the metabolism, so that can be handled both in the presence and in the absence of oxygen, they performed two parallel routes of isolation. So one that was under strictly anaerobic conditions and one under aerobic conditions. And then, after a few rounds, they looked for those strains that were present into both isolates. And they found a strain of Geobacillus thermodenitrificans, which is called, that is a facultative anaerobe, so can be grown in the presence of oxygen, but also anaerobically. And they showed that in the absence of oxygen, they could use this organism to co-consume glucose and xylose, which are two of the products of lignin degradation, and ferment it to lactate. So that, I think, was a very nice example, again, of bioprospecting, where they could isolate a good host. On top of this, when the guys performed this isolation, it was this rush and golden age of CRISPR, where a lot of CRISPR tools were developed. And these were the first isolates that happened in that period of bacteria that could, in principle, have defense mechanisms that could be comparable, with the development of a CRISPR tool. So what the same lab did was to check if this isolated bug had a Cas9 protein. And actually they found out that there was. And they did a follow-up work, very nice, where they characterized a thermophilic variant of Cas9. They isolated it, they showed that it worked, and they demonstrated that it can be used also to make gene deletions in extremophiles. And therefore, also since that moment, extremophiles had also their own Cas9 toolkit. So this is just to mention that, you know, these bioprospected strains can really be a gold mine for many applications, even beyond the original scope they were intended for. So this also, there is the link available. The second example, you know, of valorization of lignin that I wanted to touch is about expanding the acceptance range of substrates for yeast. So yeast is really a power horse for industrial microbiology. It's very resistant. It can really withstand a lot of stresses. So it's really a very good model organism to work with. However, it can only grow glucose. But as we mentioned, there is also a lot of xylose present in this lignocellulosic biomass. So a guy in the Netherlands called Jack Pronk, who actually this year received a very big prize for this research from Novozymes, decided to look at shit in literal sense. So started to think of, okay, if we want to make yeast capable of eating lignocellulosic biomass, let's look at those sources where, you know, lignocellulose is degraded. And he looked at big mammals like elephant and thought, okay, elephant really can eat branches and can, you know, get their energy from that, from trees. So they might have in their gut something that allows them to degrade this complex lignocellulosic structure. And in fact, they found out that in the gut of the elephant, there was a fungi that is capable of directly assimilating xylose. So from the poop of the elephant, they isolated this fungi, and the specific gene that was needed, called xylose isomerase, and this one was added into yeast. And what they proved is that this new yeast was then capable of consuming xylose. What they tried to do then, and I think that was really a clever approach, was to start evolving yeast to co-consume xylose together with glucose, so that then the organism could assimilate both substrates at the same time and, you know, create a real robust cell factory for the bioconversion of lignocellulose. And with yeast, you can make bioethanol. So actually what they managed to do as a consequence of this study was to create the first large facility for the production of second generation bioethanol. And it's called second generation because it comes from second generation feedstocks, so lignin derived. And this is how the process looks like. So there is the feedstock. This is like, you know, rest from, you know, wheat or corn cultivations. They are pre-treated. Then there is the addition of these enzymes for the degradation of the lignin. And then this hydrolyzate is then added into a fermentation reactor where we have yeast that is present there. And then there is the whole downstream processing to extract the bioethanol, and then you have pure bioethanol. There was a review coming out in 2017 that really showed how this second generation bioethanol started to really ramp up. These are plants that were built in the Midwest of the US, where you have a lot of corn production, so a lot of agricultural waste. And one thing that was interesting from a lecture that Jack gave was to show that when they started scaling up this cultivation, they started observing some problems that had nothing to do with science, but that, you know, you need to also face when you develop new processes. that was the presence of big stones into the bioreactor that was, transferred because of, all this agricultural waste is moved in a bulk way from one place to the other. And so, yeah, these stones, long story short, really ruin these steel tank reactors. And this had, of course, costs to be taken into account, but it's just to show that, when you try to develop these biotechnological processes, there are a lot of things you need to take into account. And these stones were pretty big. Okay, so we were talking about second generation biofuels, and now I would like to mention an example that I think is really, really a cool example of, synthetic biology to the extreme, which is the valorization of CO2 through, these fourth generation feedstocks. So what you see here is a cartoon that shows what we were discussing before, this power to X pipeline. So it means like electrical power to product. And this can really be the biomanufacturing of the future. And this is also something where I personally, I'm investing a lot of time in research. And we are trying to develop new processes where we do not use plant biomass anymore, but we really just rely on CO2. And you need to think that CO2 is the most inert molecule possible. It's completely oxidized, doesn't have any energy inside. But if we develop good electrolyzers, we can actually power up this CO2, bringing electrons into the molecule. So a very sustainable, vision of this would be having some sources of energies that come from, renewable pipelines. So maybe like solar power, wind power, or hydropower. They generate electricity, and we use this electricity as a source of electrons. And with that, we can, for example, reduce CO2 to formic acid, which is basically CO2 with 2 electrons inside. Or we can also create just what we would call sources of energy, so like hydrogen from water splitting. And then the idea is that we use these molecules as carbon or energy source to feed microbial cultivations. So we need to become very good at teaching microorganisms to efficiently use CO2, hydrogen, or, for example, formate, and convert it into, value-added compounds that could be, from food and feed to make chemicals or fuels. So one way to do this is, for example, by taking a model organism like E. coli and teaching E. coli how to grow as a plant, meaning that what it means would be growing directly on CO2. And this can be done by implementing the Calvin cycle, which is the pathway that plants use to assimilate CO2, into Escherichia coli. And you should imagine this as a kind of heart transplant. It's a very, very challenging thing to do. It means changing completely the trophic mode of an organism. If you remember this scheme we saw at the beginning of the class, it would mean you take this organism and bam, we paste it on a completely different position on the board. But I will show you that this is possible now with synthetic biology. So one way to force E. coli to grow on CO2 is the one of creating, of exploiting what we call growth couple selection. So growth couple selection is a tool that we can use. And how does it work? We take the organism of interest, so in this case E. coli, and we interrupt its metabolic network. And we do this by gene deletions. And what we do is that we take we should imagine the chart with all the metabolic reactions that are present in E. coli. By doing some gene deletions, we isolate parts of the metabolism. And what you can see here is that we need to feed external nutrients to supplement growth. However, when we introduce heterologous enzymes that can reconnect this network in different ways, we can replenish growth. And we can do it for a wide range of purposes that you can see here. In the case we're interested in, it will be a new route for the assimilation of a feedstock. So in this case will be CO2 assimilation. And the idea is that we reconnect these modules, and then looking at how good is the organism at restoring growth, we can understand, okay, the module we introduced is good. It can carry enough flux. And we can also test variants, different modules, and see which one behaves best. Again, we can use growth as a proxy, we say, to prototype the pathway and say, okay, this variant rescues growth best. So this is the pathway we should stick to, or the model variant we should stick to. And of course, since growth is the readout of how the pathway works, we can use this ability to grow as a selective pressure to do adaptive laboratory evolution to enhance even more the capacity of the pathway. So this is a very handy tool that we can use. So what you see here is how the Calvin cycle looks like. And what is important to know is that all these genes that are listed in black are natively present into E. coli, but the ones that are in green, so PRK and Rubisco, they are not present. So they need to be introduced heterologously. So we can use growth coupling as a way to select for good PRK and Rubisco variants. And the first way in which Rubisco was used was as a detoxification system to remove the product of PRK, which is this molecule here, ribulose bisphosphate, which, because of the presence of two phosphates, is quite toxic. So the idea is that if you have a good Rubisco, you remove this toxicity and you have growth. A second way, but let's put it like this. If we use this system, the whole biomass that will depend from Rubisco is very tiny, just 1%. On the other hand, if we block the classical glycolysis that you see here by making a gene deletion, we enter into this situation with the interruption of the metabolic network I was talking about before, and we can use Rubisco as a shunt to restore growth. In this way, 15% of the biomass will depend on CO2 fixation. A further way to do this is to completely split upper and lower metabolism by moving the deletion below the product of Rubisco. So in this way, we can have the whole upper metabolism that runs on CO2 fixation. And then the lower metabolism that uses an additional carbon source like pyruvate to make energy and everything that is needed below the deletion. And in this way, 30% of the biomass will be made by CO2. And finally, once you have a good module that that works well, we can select and have the whole biomass that depends on CO2 fixation. So these steps that I'm showing here seems easy, but they require a lot of time. And this is, I will show it really briefly. So here is how this group managed to create a hemi-autotroph E. coli, where only the upper metabolism depends on CO2 and the lower metabolism depends on pyruvate. So they started having, Rubisco and PRK engineered, and they started by feeding silos. And, this Rubisco should work as a shunt to fix CO2. And they started having a lot of silos in the medium. And then they started what is called a chemostat, which is a bioreactor with continuous flow in and out. So you have always fresh medium that comes in and always fresh, like spent medium that goes out. And in this system, the cells are in constant exponential growth. So they keep on dividing. And when you divide, you accumulate mutations. And what they did was, through time, they started removing more and more xylose from the medium until the point there was only CO2. Of course, they kept on feeding pyruvate to fulfill what is the requirement for the lower part of the metabolic network, but they ended up having a strain that was capable of growing on CO2 for the upper metabolism and on pyruvate for the lower metabolism. And this, they were really able to characterize it. So you can see here a growth curve of the evolved strain that can grow at high CO2 when only pyruvate is present. And the ancestor strain before evolution cannot grow only on pyruvate. So that was a very cool, let's say, checkpoint. And then the same group continued with a new design where xylose and CO2 were already, again, used since the very beginning, and formate was used for energy. And then throughout a constant cultivation that lasted, I think, half a year, they managed to emerge with this phenotype where you have complete autotrophic growth. And all the biomass comes from CO2, and formate is used as a way to create NADH that can be used to create ATP to support the autotrophic regime. So this was really a huge breakthrough that showed how you can convert E. coli from an heterotrophic organism into an autotrophic organism. And I think it's a really, really, really great work. Of course, they manage also to prove that all the biomass depends on CO2 by doing a labeling experiment where they put CO2 that is heavier, that has one extra a neutron in the atom of carbon. And with this system, they looked at all the proteinogenic amino acids, and they could show that the whole biomass was really coming from CO2. So that was the way to go. Then I will just now briefly touch on the last two examples because I think also time is running out. So another application of microorganisms that we can have is on bioremediation. So you are aware that there is a lot of trash also around, for example, in the ocean. This Great Pacific patch, garbage patch, is as big as African countries of the size of Sudan, Chad, Angola, Niger, and Libya. So it's really big. But there are now organisms that have been discovered that are capable of slowly degrading PET, which is this polymer that is the main constituent of plastic. And this PET degrades, is degraded into ethylene glycol and terephthalic acid. And especially ethylene glycol can be used as feedstock for microbial cultivation. So there could be, in principle, ways also to couple the degrading of plastic to growth of microbes, a bit as for the example we've just seen for CO2. And there is a whole vision on creating a circular economy that is based on PET biodegradation. There is a organism that is now considered the platform to study for this application, which is called Ideonella saccharinensis. But I don't know more about this, unfortunately. But it's just, there is a lot of research now and funds that are now directed for this type of application. The last example I wanted to show is the one of how we can use microorganisms to produce drugs. So there is this example that you see here that is pretty recent. So this molecule that you see here is called vinblastine and is an anti-cancer drug that is used as chemotherapeutics. And it's a molecule that is present in nature in just one species of plant that is in Madagascar, called Catharanthus roseus. And this plant produces this molecule, but in order to have 1 gram of this molecule, you need to process 2 tons of biomass. So you realize it is not a sustainable way to produce such compound. So why I'm showing this example is just to highlight that this is it came out at the end of 2022, and back then was the most comprehensive work on strain engineering for the production of a molecule. So these scientists managed to create a yeast variant with 556 genetic modifications and the heterologous expression of 34 genes taken from these plants in Madagascar. And they managed to reach these last two steps of the pathway. So catharanthine and vindoline, which then can be combined semi-synthetically together to make vinblastine. And they managed to produce quite low amounts. We're talking of micrograms per liter, but you realize that, if you can produce this with a kilogram of sugar, is much better than, processing 2 tons of the other molecule. So, one application of bugs of industrial biotechnology is also to make this very kind of niche market compounds that have a lot of value, but that would have a high environmental cost if we would have to extract them from natural resources. So with this, let's say we have concluded. I just wanted to make a question. So you have seen now some example of top-down, and let's say we discussed what top-down and bottom-up approach means. You have seen now 5 examples, and I would like you guys to start also maybe writing in the chat. If, for example, you think that, the first example we discussed, so isolation of thermophiles for lignin valorization, is a top-down or a bottom-up approach for synthetic biology. And I have few in the list. So there is first the isolation of thermophiles. Second is the example of engineering yeast to enable co-consumption of xylose and glucose. Is this top-down or bottom-up? Third example, enable growth of E. coli via the Calvin cycle. And then the use of Idionella for PET degradation. And the last example is building vinblastine with yeast. So if you can write the 4 numbers, and A for top-down and B for bottom-up, I think I give you 2 minutes, and then we can look at the result. Okay, so I see the examples for number 1, but you can just start write for all of them. So take your time. I can give you 2 minutes, and then we comment. Okay, so yes, I see people are still writing. So I think if we are done with that, I can maybe show the results. So these are the solutions. So I see, for example, Maxwell got them right, and I think also Abigail. So indeed, the examples of the isolation of the thermophiles and the use of this Idionella are examples of a top-down approach you can have in synthetic biology. And the other 3 examples, so yeast to express the xylose isomerase, the heart transplant of E. coli to grow on the Calvin cycle, and the expression of the vinblastine pathway, those are examples of bottom-up synthetic biology. So with this, let's say we are at the end of the class. So I hope I managed to convince you that there is a wide range of microbial diversity that exists in nature. And those traits that are advantages for us, we can exploit, and we can try to build some, bioprocesses for a bioeconomy. And we can try to enhance these valuable traits, using combined top-down or bottom-up approaches. And of course, top-down and bottom-up do not exclude each other. You can also have some isolates that are very good, and then eventually you have to do something bottom-up also with them. But the examples I showed, let's say, were, I think, clear examples of one or the other. And yeah, I really hope that you are convinced that microbial applications can contribute to, supporting the implementation of the sustainable developmental goals that our society needs. And with this, I conclude the class. So here are my contacts in case you have also questions for the future. And I'm now happy also to, if you guys have more general questions, or if there were some questions from the chat, I think I can now take time to answer them. Thank you very much. So let's now stop maybe sharing. I will stop sharing now, so if I can see better. Okay, so if there are no questions, as I said, feel free to unmute and ask things. And in the meanwhile, yeah.
Just a little curious.
Yes.
They use the valorization of carbon dioxide. Doing it from E. coli and doing all those edits. Is it not spending where you wouldn't have spent by just simply using, for example, cyanobacteria to be able to do that, which already has the requisite enzymes to be able to do that?
Yeah.
What triggered you, or what encouraged you to think of E. coli instead of, please?
So I think this is a great question, Billy. So thanks for asking. Indeed, I would say if you would have to make a bioprocess out of it, I agree with you that E. coli is not probably the best organism to pick. And I would agree that in nature, there are already better ones. I think we should look at this as more of an academic exercise that tries to show that you can really rewire the metabolism of a given organism in a way that was unprecedented. And in principle, if you are able to find other organisms that have some sort of other features, for example, I give an example. You want to produce something that, I don't know, you have a strain that is very good at making a polymer. And the pathway to make this polymer is not easy to express in a cyanobacterium. It might be easier maybe to teach this organism to grow on CO2 and make this polymer from CO2. So I agree with you that E. coli is not maybe the best example for an application, but I think that this paper shows you that in principle, one organism that is not capable of growing on CO2 can become an autotroph. And then I think you should take this as a learning kind of outcome and say, okay, then when I will find the organism that is good at what I need, I could try to teach him to grow on CO2.
Thank you very much. You've really answered my question, and I want to comment you. I enjoyed the lecture from the word go. Thanks a lot.
Okay.
Yeah.
Okay. Thank you, Billy. Happy to also have the chance to speak with you guys. So I see some questions. So Nelson was asking if we can find microalgae in deep waters. I would say that depends what deep means, because you should imagine that the lower you go into the surface, the darker it becomes. So I think at the bottom of the oceans, there is no light, so we will not find microalgae there. Then I see, so Jeremy was asking, would it be possible to replicate some of the system in Archaea for more versatility in extreme environments? So I'm not sure what we were talking about when you asked this question. So I guess about microalgae. So probably was in the part on microalgae. Well, the point is that microalgae, especially if you are interested in food applications, microalgae already are kind of considered as food and safe to be used as food. I think you would have to consider what are the regulations there, and I would imagine there is not a lot of freedom to propose Archaea as an ingredient, whereas you have it, for example, spirulina is used a lot as an ingredient, also for nutrition. So algae, I think, are a bit better candidates for that. I see Murimi was saying, okay, cool idea, maybe proteins and other structures that allow for extremophile to survive in such condition can be replicated. But for example, there are, as we saw, some extremophilic, heat-tolerant microalgae. I know that there are also pH-tolerant microalgae. So definitely there is the possibility of apply some concept of extremophiles and enhance some traits of algae that relate to the extreme conditions without having to move to Archaea. That is for sure possible. I wonder what is the level of each nutrient content in this product. So Nelson was asking this. I don't know what we were talking about. So I'm afraid I cannot reply now. So yeah, Rubisco. Yeah, I think the questions in the chat. So Miriumi, question on a previous discussion we were having above. What would take to transfer the traits of an extremophile to other organisms? I think it's very difficult. So I think in general, it's much easier to take an extremophile and then engineer it, bringing other traits, because I think something like resistance to high temperature or low pH is something that is very difficult to engineer in a model strain, I would say. I think it's actually a very, very good question. Yeah, so I do not see other questions in the chat. If someone has also curiosities and things, feel free to ask. Well, if not, I think then we reached the end of today. Before I forget, actually, I should have said this maybe at the beginning, and I think I didn't. So I apologize for that. But let's say we are reaching also the time to work on the projects. So there are some updates from Heidi and the organizing committee. So anyone who didn't get a group assignment yet, should receive it by the end of today. Keep in mind Boston time as a timeline. And everyone who submitted the project idea will get feedback and approval also today. And you should, let's say, the only project you can present is a project that has been approved at this step. And next week, there will be the meeting during the normal lecture time, but there will be the chance to be divided into breakout rooms for each group to discuss their projects. So this was from Heidi. From my side, I do not have other things to add. I really hope, guys, you enjoyed and you managed to learn something new. And yeah, as Nelson also suggested, if there are some other questions on Slack, yeah, please let me know, and I will be happy to help. Okay.
Okay. Thank you so much, Enrico, for such a wonderful lecture. And I would like to also remind you that we are organizing a webinar event next week on Friday, on 21st of June. We shall have Alex Izzo, the chairman and CEO of Gojek. Gojek is a competition for students and other people from all over the world. It's about synthetic biology. And what it helps people to participate free of charge and online. You don't invest anything. So please, I invite you to attend and listen to what Alex will be talking about, and you learn more about Gojek. You never know the projects which you're doing as final projects for SynBio4All. You can transfer them into the competition for Gojek. Thank you so much. And I really appreciate Enrico for such a wonderful and engaging lecture. Thank you so much. And that's what I had to say. Thank you.
Thank you, Emma. And have a good night, everyone. Bye-bye.
Bye-bye.
Ciao, ciao.
Bye-bye. Ciao.
Ciao, ciao.
Good lecture, yeah.
Thank you. Bye.